Today we're continuing on with our look at the different categories of organisms that might cause us harm. Last day we looked at the prokaryotes, the bacteria. Again, archaea are not harmful to us. Today we're going to look at microscopic eukaryotes. So I've put simple eukaryotes here. Remember, any kind of cell really isn't all that simple, but we're looking at microscopic eukaryotes, the protists, and also the fungi. Now, fungi are not protists. In fact, fungi are closely related to animals, but I'm going to cover the eukaryotic parasites in two groups, protists and fungi today, and then in the next topic we'll look at the multicellular animal parasites, which of course are things like tapeworms. Let's start with a bit of review, because review is always a good thing. Remember that prokaryotes and eukaryotes differ in a number of fundamental ways, most of them related to their basic structure. Prokaryotes don't have a nucleus. They don't have the complex organelles and compartments that we see in eukaryotes. They also reproduce exclusively asexually. They reproduce by a process known as binary fission, a simple splitting of the cell. This is a rather rapid method of making copies. Now remember that sex is defined by the process whereby two parents each donate a full set of information to the production of a brand new individual. In prokaryotes, we do have the exchange of information. It's a lateral exchange in that two cells will come together, they'll swap bits of DNA, and they'll both change potentially in the process, but we don't get a brand new individual that's offspring of the two starting individuals. In eukaryotes, we have two types of division. We have asexual, this is mitosis, and when we're looking at microscopic eukaryotes, this is exemplified by budding, which we'll talk about at some point. This is where one cell kind of splits off of another cell. The production of spores can also be asexual. Spores can also be produced sexually, though, as we'll also see. In sexual reproduction, so this is a result of meiosis, we have the combination of genomes from two individuals. So in animals like ourselves, for instance, we have the sperm and the egg. They each contain one set of information. The information comes together to produce a diploid organism, a brand new organism that has two sets of information. There's some variation as to how that happens in different eukaryotes, but the basic concept here with sex is that sex is always due to the contribution of two parents generating a brand new individual. A typical eukaryotic cell here, we have a nuclear envelope that consists of two lipid bilayers. And then within that, of course, we have linear chromosomes. Outside of that, within the cytoplasm, we have a number of organelles that are going to perform very specific functions. So the Golgi apparatus, the smooth ER, the rough ER. We have mitochondria, which, as we've discussed, are derived from bacteria. They contain their own DNA, but a big circle of DNA. We have a flagellum, perhaps, or we might have cilia, or we might have none of these motility-related structures whatsoever. Remember that the flagella and the cilia are quite different from the flagella that we see in bacteria. They don't spin, instead they wave from side to side. The eukarya is made up of four kingdoms. We have the plantae, we have the animalia, we have the fungi, and then we have the protista. Out of convenience, we can divide the protista into three informal categories. So these are not proper taxonomic categories, but these are three groups that are defined based on how these organisms feed themselves, how they acquire nutrients. So first of all, we have the protozoa. These critters are animal-like. They tend to be motile, although not all of them are, but they tend to move around, which is something we kind of associate with animals. Animated, that word comes from animals. I mean, a cartoon where there was no movement would be a rather boring cartoon, but animals tend to be things that move around quite a lot, although there are exceptions. So the protozoa, they're animal-like, they tend to move around, they gobble up food, they ingest it, they take it in big chunks, and we have at least 7 phyla. A phylum is a large group of organisms. We have the algae, which are plant-like. We're not going to focus too much on them because, of course, they're not pathogenic, they're making their own food. Why would they want to parasitize something else? Then we have the water molds, and these are fungus-like. These are single-celled, and some multicellular examples exist as well, but these are organisms that will secrete enzymes into their surroundings, break food down outside of their own cells, and then absorb those simpler, smaller nutrient molecules. Now we're also going to cover fungi, which would be the molds and the yeasts and the mushrooms, but realize that fungi are not protists. I've just grouped them into this lecture because it kind of makes sense. They're simpler eukaryotes, simpler than the multicellular animal eukaryotes that we're going to talk about, and we're going to look at things that are, for the most part, microscopic in this topic. This is a remarkably simplified view of how the major groups of organisms are related to each other, but it should suffice for our purposes. Remember that bacteria and archaea are both prokaryotes, and that group is based entirely on their structural similarities. They do differ quite a bit when it comes to their biochemistry. Bacteria and archaea appeared very, very early in the Earth's history. Essentially, once the crust had cooled, life started to appear, and these are the two groups that cropped up. The eukarya are an offshoot of the archaea. They share a lot of biochemistry with the archaea. And the eukarya can be divided into 4 kingdoms. We had the eukarya diverging quite early on into these different groups. We had the plantae, we had the fungi, we had the animalia, and these are all groups that are fairly well defined. But during that splitting process, a lot of these early groups kind of went their own way. And we define that massive diversity as the protists. So we have all these very diverse organisms that we can't fit into the plants, we can't fit into the fungi, we can't fit into the animalia, and they've all been stuck into this category of leftovers, which we refer to as the protists. So for instance, we have members within the Protista that will act like plants if there's sunlight. If there's sunlight, they have chloroplasts. They will use them to collect solar energy, and they will use that energy to make their own food. But if you turn off the lights, they start to act almost like amoeba, and they start to gobble things up, which is something we associate with animalia. So the Protista group is quite fascinating. It's not really a technical taxonomic group in that a lot of the organisms in that group are not closely related to each other. For the most part, they're organisms that we can't figure out what to do with. We can't figure out how to classify them. In terms of their fundamental biology, the protists are the most diverse group of the eukaryotes. They have lots of different strategies when it comes to feeding themselves and having sex and all the other things that living cells need to do. The majority are unicellular, so they're single-celled, free-living critters. Some of them are colonial. A colony of cells is a situation where lots of cells come together and they benefit from being together, but at the same time, you could pull them apart and they could survive on their own. A number of the seaweeds are quite large and they're multicellular. Multicellular organisms consist of cells that can't survive on their own, and typically the cells are specialized. We have some cells that are involved in absorbing nutrients, we have some cells that are involved in reproduction, etc. And we see that within the large multicellular protists. The majority of protists are motile. They use flagella or cilia to move around. In many cases, the adult form is sessile. That means it doesn't move around, but they have gametes that can move. They have complex life cycles in many cases. We have an alternation of generations in some of the more advanced forms, the seaweeds. They can alternate between sexual and asexual reproduction as well. This is often the case for a lot of organisms. If conditions are good, they'll reproduce asexually because it's fast. If conditions are bad, they reproduce sexually, and that results in offspring that are all different. And hopefully next season, when conditions become good again, we'll have selection for some of the organisms that, are best suited for that particular environment. That's an idea we'll come back to. Some of them will form cysts, these very resistant cells that act as spores. They're similar to endospores. They live in a diversity of habitats, and they have very diverse nutritional requirements and nutritional abilities. Many of them, the majority, are free living, which means they're not feeding off of something else. They're going their own way, but there are a few that are parasites, and again, of course, that will be our focus. And then there are some that are commensalists, that live on other organisms but don't harm them. We have our three informal groups of protists, and I have to stress that these are informal groups. They're not proper taxonomic groups, but they're convenient, especially for us in this course. We have animal-like Protista. They're animal-like because they're ingestive. Remember, ingestion refers to taking in food in large chunks and breaking it down within your body. Or if we're talking about single-celled Protista, the breakdown occurs within the cell. So within this group, we have the amoeba, we have the ciliophora, which would include things like paramecia, so cells that are typically covered in cilia. And then we have a few parasitic forms, which of course is what we're going to focus on. We have things like Trypanosoma and Giardia that we'll get back to. Next, we have the photosynthetic protists, and these are informally referred to as the algae, and also the seaweeds. So we have Rhodophyta, the red algae. If you enjoy sushi, sushi rolls are wrapped in red algae. We have the green algae, we have the brown algae, we have the golden algae. There's a lot of cool stuff here, but we're not going to talk about it very much because these are not parasitic groups. They're manufacturing their own food. They do not need to parasitize other organisms. And then finally we have the water molds. These are Protista that resemble fungus in a lot of ways. They are absorptive. So what they do is they release exoenzymes out into their surrounding environment. That breaks down their food outside of their cells, and then they soak up amino acids and nucleotides and these simpler, smaller molecules. There are some forms of the water molds that do cause us problems. Recall that motile, M-O-T-I-L-E, means capable of movement. There are some eukaryotes that are incapable of movement. They're non-motile, but the majority are capable of movement or have specialized cells that are capable of movement. The movement is provided by flagella or cilia. These structures are different from what we see in bacteria in that they wave back and forth instead of spinning. We don't have an electric motor at the base of a flagellum or cilium. But cilia and flagella themselves are very, very similar in their structure. Their structure is identical. The only difference is size. Essentially, cilia are small flagella. Eukaryotic cells will tend to have a small number of flagella, or they'll have a lot of cilia, and the cilia may cover the entire surface of the cell, as you're seeing in the bottom diagram here of a paramecium. In eukaryotes, we have this complex structure made up of microtubules. Microtubules are made up of smaller proteins, but they're these hollow tubes that serve a structural function, but they can also slide past each other. We have what's called a 9+2 arrangement. We have two microtubules within the middle of the flagellum or cilia that offer support, and then they're connected by this radiating structure of proteins to doublets of microtubules that are found around the circumference of the flagellum or cilia. So a doublet consists of two microtubules stuck together. We have 9 of these doublets. In between the doublets, we have motor proteins that can change shape if you supply them with ATP. And when they change shape, they cause this ratcheting motion whereby the doublets slide past each other. And that causes the entire flagellum or cilium to bend. And it bends back and forth, waves back and forth, and brings about movement. This entire apparatus is surrounded by a cytoplasmic membrane, a plasma membrane that's continuous with the plasma membrane of the cell. So this is a rather thick structure. This is something you can see under the light microscope. If we compare that to what happens in prokaryotes, as we've talked about, we have this really neat electric motor that's embedded within the cell wall and the plasma membrane. We have hydrogen protons that will flow through this rotor and cause it to spin. It's an electrical motor, which is really, really cool. And it will spin, and it causes the flagellum to spin like a propeller. The flagellum is made up just of protein. There's no cell wall or plasma membrane surrounding it. That means it's a much thinner structure. So we have two very different strategies here for moving the cell, and they're both pretty darn complex and pretty darn cool. The other thing to note is that all eukaryotes that have cilia or flagella have the exact same structure. It doesn't matter if we're talking about protists, if we're talking about human sperm cells, if we're talking about the cells that line the trachea that have cilia, they all have the same fundamental structure, presumably because this structure appeared very early on in the evolution of eukaryotes, and all the descendants of that ancestral cell share this structure. And here are some pretty typical examples of motile unicellular protists. We can have a single flagellum or a small number of flagella, or alternatively, we might have a whole lot of cilia that coat the entire cell. Again, though, the structure of the two, cilia and flagella, are identical. Cilia are simply smaller and usually more numerous. Several of the Protista can form very resilient cysts that are similar to the endospores formed by gram-positive bacteria. Remember that gram-positive bacteria, if they're faced with an adverse environment, instead of fighting it out, what they'll do is they'll form this dormant endospore, and they'll just wait it out, and when things get better, they can germinate and go about their business. There are a number of protozoa that do the same thing. So for instance, if you take some straw or hay, put it into a jar of water, you'll notice within a few days that we get a lot of protozoa coming out and becoming very active. They came from cysts that were on the grass. So as conditions dried out on the grass, they formed this tough outer coating that protected them and allowed them to enter into a dormant state. A number of protozoa that are parasitic will use this cyst to bypass the high acidity of the stomach. So for instance, Giardia. If you get Giardia, what you've done is you've ingested a cyst that has passed through the stomach, and then once it gets to the gentler environment of the intestine, the cell is going to break out and go on with its life cycle. So we have these free-living, active feeding stages known as trophozoites. If the environment becomes adverse, they can form a cyst. The cyst allows this cell to remain dormant for long periods of time, and then when the environment improves, they can exit the cyst and become active cells again. The classification of the Protista has changed quite a lot over the last few decades. And again, that's because we've taken a lot of organisms that aren't necessarily closely related and thrown them together. As we find more evidence to support relationships, we get a better picture of what's going on within this group, but it will continue to change until we sort out all the specifics of these relationships. If you look at different textbooks, you'll find different representations of the subgroups within Protista. I've chosen this diagram because it's a good kind of consensus. I think most people would be okay with this classification scheme, and it does a nice job of pointing out some of the groups that contain pathogens. I have added arrows to show you which groups contain protists that are potentially harmful to us. So first we have the Excavata. All of the subgroups, the large subgroups within Excavata, contain at least some representatives that are potentially pathogenic. Next we have the chrome Alveolata, and we have a few groups here that can cause us harm. We have the Dinoflagellates, which are not parasitic, but I've included them because some of them do produce a toxic waste product, which can cause shellfish poisoning. The Apicomplexans are all parasitic. That's kind of unusual within the Protista, but this group consists of organisms that are highly evolved. They have specialized structures that allow them to penetrate other cells and parasitize those cells. They become endoparasites. The ciliates are by far non-pathogenic, but there are a couple of species that might cause us problems. In the middle here, the Rhizaria and the Archaeplastida, we're not going to talk about them at all. These are creatures that don't contain any pathogenic members. Note that they've included the land plants there just to show how they're related to these green algae. Land plants are an offshoot of early green algae. Then we have the Uniconta. Uni means one, conta refers to the fact that these organisms, at least in some part of their life cycle, have a single flagellum. Notice that fungi and also animals are contained within the Uniconta. So in us, in humans, we only have one cell that has a flagellum. That is the sperm cell, but it has one flagellum, which does indicate some relatedness to these groups, and we'll come back to that when we talk about the fungi. But we have one group here, the Entamoeba, which are amoebas that can parasitize the digestive system. Okay, let's take a look at the first group of Protista that contain pathogenic members. The Excavata is subdivided into three groups. We have the Diplomonadida, the Parabasala, and also the Euglenozoans. The Diplomonadida and Parabasala are very unusual in that, unlike any other eukaryotic cells, they lack mitochondria. Remember how useful mitochondria are. Mitochondria allow us to get a whole lot more energy out of our food, and they also absorb oxygen. Oxygen is very helpful, of course, but it can damage cells. It's very reactive. It can oxidize parts of the cells and cause damage. For that reason, these two groups, Diplomonadida and Parabasala, are found in areas that lack oxygen. They're anaerobic. They're actually poisoned by oxygen. So they live in areas where there's not a lot of fresh air available, as we'll see. The Diplomonadida contain multiple flagella and two separate nuclei. And a good example of this group would be Giardia. Parabasala, on the other hand, they have flagella, but they also have a flagellum that is strapped to the side of the cell by a membrane, an extension of the plasma membrane. And this forms almost like a fin going the length of the cell. You might have seen clownfish or eelfish before that have a fin that runs the entire underside of the fish, and it waves back and forth and causes movement. Well, the same kind of thing is happening here. And the best example of this would be Trichomonas vaginalis. You can probably guess where you would find this critter. Now, I should point out that no one's quite sure why they don't have mitochondria. It was originally thought that this is a very ancient group of eukaryotes. Maybe they evolved before eukaryotes took in bacteria as mitochondria, although recently they have found evidence for bits of leftover mitochondrial DNA. So it seems that they did have mitochondria at one point, but they lost them a long, long time ago. So we have the Diplomonadida, we have multiple flagella, we have 2 nuclei, and they look like eyes when you're looking at them under the microscope. And underneath that, we quite often have a large vacuole. That's what's making the smiley face. Then we have Parabasala, and Parabasala, again, they have flagella, but they also have this undulating membrane that they use to move around. Giardia causes a condition known as beaver fever, not to be confused with Bieber fever, although they both cause explosive diarrhea. Anyway, you acquire a Giardia infection by drinking contaminated water. This is a protist that inhabits the intestine, and occasionally it will produce these very resistant cysts that are passed in the feces. If the feces end up in the water, then that contaminates the water, and someone else might drink some of that water, take in those cysts, and themselves become infected. The cysts can resist the passage of the cell through the stomach. The stomach is incredibly acidic, and it tends to denature and break down most proteins. But these cysts can survive that trip, and then they'll germinate within the small intestine. Trichomonas vaginalis is the only protist we know of that causes an STD, a sexually transmitted disease, in humans. Males are usually asymptomatic, but they can transfer Trichomonas vaginalis to a female. In females, the most common symptom that we're going to see is a thick discharge from the vagina. That's where they're going to spend their time, and also a fishy odor. This may be the most common pathogenic protozoan infection in humans, and in fact, it might be the most common STI in humans. The last major subdivision of the Excavata is the Euglenozoa. Now, unlike Parabasala and Diplomonadida, these guys do have mitochondria, and I should point out that the vast majority of the Euglenozoa are not pathogenic. For instance, Euglena, which is a fascinating little photosynthetic organism that you can find in local ponds and lakes and so on, is not a problem to us. But within the Euglenozoa, we have a smaller subgroup known as the Kinetoplastida, and these guys are pathogenic. They're quite fascinating in that they have a huge mitochondria. They have this big swollen mitochondria that is visible quite easily with a light microscope. So the mitochondria contains a lot of mitochondrial DNA. So they have extra sets of mitochondrial DNA. That DNA is all condensed into a darkly staining region known as the kinetoplast. Let's take a look at a couple pathogenic members of this group. A really important genus is Trypanosoma. This contains a number of different blood parasites. We have one that causes African sleeping sickness, one that causes Chagas disease, and we'll come back to those. In the photograph on the bottom left, you're seeing a sample of contaminated blood. The round cells are red blood cells, and the squiggly cells, of course, are the Trypanosoma cells. Note that within the Trypanosoma cells, we have a very dark staining area, which you might think is the nucleus. Actually, it's the kinetoplast. That massive mitochondria contains a lot of DNA that takes up stain quite readily. In the upper right, you're seeing contaminated blood as viewed through the scanning electron microscope, so we can see it in 3 dimensions. Again, those round kind of donut-shaped cells are red blood cells, and the squiggly cells are the pathogen. Trypanosoma are transferred from one host to another by an insect vector. So a biological vector is an organism that can be infected by a pathogen, and also pass that pathogen onto another host. So at the top here on the right, we have the tsetse fly, which is a vector for Trypanosoma brucei, which causes the African sleeping sickness. So if we want to control the spread of this particular pathogen, we have a few targets that we can go after. We can go after the pathogen itself, but we can also try to control the fly population, or something even simpler, put up netting when you're sleeping so you're less likely to be bit by this fly. The other insect that you're seeing is something known as a kissing bug. Now, it's an insect that has kind of a hypodermic needle attached to its face that it can draw fluids through. And what it likes to do is it likes to draw blood from sleeping humans. We have nice, soft, supple lips, and that's where it tends to get its blood from. So as you're sleeping, this guy might crawl into your face, suck some blood from your lips, and in the process, transfer some trypanosoma. Now, I do want to make sure you realize that if you see a bug like this here, where we live, it's not the right type of bug. It's probably something called an assassin bug, which might be living under your clothes dryer. It uses that needle to suck the guts out of other insects. You don't have to worry about those guys. Here's a fascinating and non-pathogenic member of the Euglenozoa. This is Euglena. It's a photosynthetic organism. It contains chloroplasts, and it will harness the energy from the sun to make food. It has a red spot there called the stigmata or eye spot that it uses to sense light, and it moves towards the light. If you put it in the dark, it acts like an amoeba. It changes shape and it starts gobbling up other organisms. So it acts kind of like a plant, but also kind of like an animal. And biologists have argued over where this critter belongs for hundreds of years. Zoologists, people that study animals, refer to it as Euglenozoa. Zoa means animal, whereas botanists, people that study plants, refer to it as Euglenophyta. Phyta means plant. The next major group that we're going to look at is the Alveolata. Alveolus means little sac, like the alveoli that you have in your lungs. Well, these guys have little tiny microscopic sacs under the plasma membrane. No one's quite sure what they do, but they appear to be involved in regulating the movement of water across the plasma membrane. We have three groups within this larger group. We have the Dinoflagellates, we have the Apicomplexa, and we have the Ciliophora. Now, in terms of pathogens, the Apicomplexa is the important group. All of these members are endoparasites. They have evolved specialized structures so that they can penetrate other cells. The dinoflagellates don't contain any parasitic members, but we will touch on them briefly because they can produce some harmful toxins. And then the Ciliophora, most of them are harmless, but there are a few species that can cause intestinal infections. Dinoflagellates are photosynthetic. They capture the energy of sunlight and use that to make food. They're part of the phytoplankton. This is a community of photosynthetic microbes floating up high in the oceanic waters, capturing sunlight and then being fed on by a lot of other things. So dinoflagellates form the basis of a number of important food chains throughout the oceans of the world. They have a really interesting structure. They're protected by an armor of cellulose plates. Cellulose is normally associated with plants. Within these cellulose plates, there are grooves that contain flagella that allow them to move around. A lot of dinoflagellates also have these really sharp spikes that help to protect them. The issue with dinoflagellates is that they occasionally go through these explosive bursts of population growth. And when they do, they can actually turn the water red. So it results in something known as a red tide. The ocean actually becomes blood red. A number of shellfish, oysters and clams and mussels, feed on dinoflagellates. And normally that's not an issue, of course, but if there's too many dinoflagellates, it can poison the flesh of these shellfish. The dinoflagellates produce a neurotoxin, as a natural waste product. So if that builds up within the shellfish and you eat the shellfish, you may become paralyzed by what's known as paralytic shellfish poisoning. It paralyzes the muscles, and that might include the diaphragm. And if your diaphragm is paralyzed, that's a bad thing, because of course you're going to have a hard time breathing. So these fascinating little photosynthetic microbes are generally not a problem. They make seafood possible, and seafood is yummy, but on occasion they can result in paralytic shellfish poisoning outbreaks. But understand that this is not a result of infection. This is poisoning. And this isn't just something that affects humans, it affects other sea life as well, such as this unfortunate whale shark. Now let's move on to an actual pathogen. Members of the Apicomplexa are exclusively parasites of animals. They have evolved structures at the apex or tip of the cell that are used to penetrate other cells, enter into the cells, and allow them to become endoparasites. These structures make up what's known as the apical complex. And we have two types of structures. We have some structures that will physically tear a hole into another cell, so they mechanically disrupt the host cell. And you can see that on the right. So down here we have this elaborate protein structure, and these proteins act as little knives. Basically, they're shot out from the tip of the cell and they punch a hole into the host cell. But we also have, as you can see on the left here, these sacs that contain digestive enzymes. So along with these mechanical shears, we have enzymes that are pumped into that space that will help soften and digest the membrane of the host cell. And here's a closer view of those structures. Once again, we have these knife-like structures that will cut holes into the cell that they're going to infect, and we have these sacs that contain these powerful digestive enzymes. Probably the most important apicomplexan in terms of human health and human history is Plasmodium. This is a protist that causes malaria. It infects mammals like ourselves, and it also infects mosquitoes, and mosquitoes serve as the vector. They transmit this protist from one mammal to the next. So let's start with the mosquito landing on a mammal, such as a human. Now at this point, Plasmodium is living within the salivary glands of the mosquito. When the mosquito bites you to draw some blood, it's going to inject Plasmodium cells. These cells are known as sporozoites. Now I'm not going to have you remember all this terminology. I just want you to know the general cycle that Plasmodium takes. The sporozoites are going to travel through the blood to the liver. They have that apical complex that allows them to penetrate cells, and they will penetrate the hepatocytes, the cells that make up the liver. Inside the liver cells, as endoparasites, they will reproduce asexually, fill up the cell will rupture and of course be damaged or destroyed in the process, and new cells will be released into the bloodstream. Those cells, again using an apical complex, will penetrate red blood cells and do the same thing. They will divide asexually within the red blood cells, fill up the red blood cells will rupture and spill out these cells into the bloodstream. So you can see that very rapidly we have an increase in the number of cells. At this point, the cells will give rise to gametocytes, male and female sex cells. These can be taken up by a new mosquito. Within the mosquito, these cells will meet up within the gut of the mosquito and give rise to zygotes. The zygotes will give rise to new mature cells that will migrate to the salivary glands of the mosquito, and this completes the life cycle. Now, as you can imagine, Apicomplexans dividing within cells and rupturing the cells can cause a lot of damage. And in malaria, you tend to go through periods of severe fever and chills. And this is where red blood cells are being ruptured at the same time. So the incubation period of these cells is the same, and you tend to go through just waves of rupturing cells that bring about these intense symptoms. In much of the world, the tropics and subtropics, malaria is a very important infection. It causes lots of death, particularly in children. Now, we're fortunate where we live in Canada in that we have harsh winters. You might not think that that's a fortunate thing, but as you can imagine, that interrupts the life cycle of the mosquito and other important vectors. There are a number of organizations that are trying to eradicate or reduce malaria. And here you're seeing projections of where they're hoping this can be accomplished in the near future. Bill Gates, incidentally, that's one of his big pet projects, is trying to eliminate malaria. One of the most important and simplest solutions to the spread of malaria is mosquito netting. So if we supply people in poorer countries that are exposed to malaria with mosquito netting that they can sleep under, this greatly reduces the number of mosquito bites, which greatly reduces the amount of Plasmodium spread. Now, this is a really good long-term solution because, of course, if fewer people have Plasmodium, then they're passing less Plasmodium to the mosquitoes. And over time, even though we're not eradicating the mosquitoes, we're reducing the parasite load in the mosquitoes, and we're reducing the spread of new Plasmodium. There are many more species of Apicomplexans out there. And I don't expect you to know these ones. It's just they are interesting creatures with interesting life cycles. Sometimes they have very, very specific needs, and they focus on very specific hosts. So if you're interested, here's a few you might want to read up on. We have Cryptosporidium. These are Apicomplexans that you can pick up from contaminated water. So if you drink water that contains the cysts of these Apicomplexans, they will germinate within your intestine and lead to watery diarrhea as they damage the cells lining the inside of the intestine. We have Besnoidia, which affects reindeer and caribou. And I thought it was kind of fitting, seeing as we live in the caribou region. You can see a poor little caribou here that has this nasty infection of the skin and loss of fur associated with that, caused by this Apicomplexan. We have Toxoplasma, which is something that can infect humans, although that's not their natural host. Usually they focus on cats, like this grumpy little fellow. Toxoplasma gondii typically infects two hosts, cats and mice. Cats are the definitive host. Now, this is a concept that we'll come back to in more detail when we talk about multicellular parasites. But a definitive host is where sexual reproduction of the parasite occurs. So within the cat, we have parasites that are producing male gametes and female gametes. They're coming together and they're producing a zygote. We refer to that zygote as an oocyst, a fertilized egg, basically. That oocyst can be picked up by an intermediate host, the mouse. Within the mouse, we have development of this parasite. The parasite may develop asexually, but there's not going to be any sex occurring within the mouse. If the mouse is eaten by a cat, then we have completion of this life cycle. So that parasite is passed to the cat. The parasite is going to reach sexual maturity, and we can have a continuation of this process. Now, instead of being consumed by the mouse, that zygote that oocyst, can be consumed by a pig or a cow and passed to a human. The human that eats that meat can then become a host. The other thing that can happen is these oocysts can be breathed in by humans that are perhaps changing cat litter. That can be a big problem if the particular human that's doing this is a pregnant female. This is one of those weird cases where this pathogen can pass through the placental barrier and also infect the fetus. The last group of the Alveolata that we'll look at are the Ciliophora, or ciliates. As the name suggests, these are cells that are covered in cilia. They use the cilia for moving around and also for feeding. Very few ciliates are pathogenic. The vast majority are free-living, they wander around, they gobble up bacteria and other bits of debris. They have two types of nuclei. They have a very large macronucleus that contains multiple copies of their genome, and then they have some small micronuclei that they might swap with other cells. It's actually a form of conjugation. The best example of this group would be the paramecia, which is what you're seeing in the diagrams here. So paramecia are covered in cilia. They use the cilia to move around. They also use it to waft food into an oral groove. And you're seeing that here. So we have food coming into this oral groove and then being packaged into a food vacuole. Anything that's useful is going to be absorbed from that food vacuole, and anything that can't be digested is going to be dumped out through exocytosis. These are really complex cells. In fact, they tend to have one area where exocytosis occurs. It's referred to as the cell anus. They also have contractile vacuoles that will remove excess water. As cells go, these are among the most complex cells in existence. They're also visible to the naked eye, just barely, but if you have good eyesight, you can see them. And here is a typical paramecium swimming around. As it rotates, you might be able to pick out the oral groove. Not all ciliates are free swimming. Some of them are sessile. They're attached to something and they stay put, but they have a ring of cilia around their oral groove. That's what you're seeing here. This is something called stentor, and it is wafting in food into its oral groove, and some poor unfortunate little ciliate has become trapped in this current and has become kind of a little hacky sack or soccer ball for this stentor. So why have I mentioned ciliates? Well, first of all, they're pretty cool. And secondly, there is one pathogenic example. Balantidium coli. This is a ciliate that can reproduce within the colon and cause diarrhea. It's typically not life-threatening. It's not a huge concern in most cases. Incidentally, maybe you've picked out the fact that coli refers to the colon. E. coli, for instance. The last group of protozoa that we'll look at are the amoebozoa. These are single-celled organisms that have an interesting way of moving around. Instead of using cilia or flagella, they produce lobes, known as pseudopodia or false feet, and then they push all of the cytoplasm into that lobe, effectively moving the cell forward. They can also produce pseudopodia to engulf food items and then ingest those food items. It's worth noting that we have cells in our body that behave this way. White blood cells can produce these lobes. They can move around using pseudopodia. And this is what that movement looks like. It's really quite hypnotic and fun to watch. Although the vast majority of amoeba are non-pathogenic, there are a few forms that can infect the body. Some of them can form very, very resistant cysts. An Entamoeba is an amoeba that can infect the intestinal tract. So entero refers to the digestive system. Balamuthia is a rather recent discovery. This is a type of amoeba that causes very rare infections. It's generally acquired by breathing in dust, breathing in contaminated soil, but it can cause severe skin lesions, it can move throughout the body, cause systemic infections, and cause a severe form of encephalitis, which is an infection of the brain. Now we're going to leave the Protista and move on to something quite different, the fungi. And remember, you can't spell fungi without fun. Fungi are a very big and diverse group. We have 20,000 known species, but there's definitely a whole lot more that we haven't identified yet. This group consists of the molds, the yeasts, the mushrooms, etc. They have a cell wall, and that cell wall is made of chitin, which is quite similar to cellulose, except it contains some nitrogen. The vast majority of fungi are decomposers. They break things down and they recycle materials. Did this joke land? Probably not. The Good, the Bad, and the Ugly? A great old flick starring Clint Eastwood, although it has nothing to do with fungus. We use fungus for lots of different things. We use it as a food item, mushrooms and truffles. We use it to generate ethanol. We use it to generate carbon dioxide that causes bread to rise, and we use it to generate a lot of food items, blue cheese, soy sauce, etc. Saccharomyces is a type of yeast that is very, very common in research laboratories. So we use it to investigate how eukaryotic cells work. We used fungi to produce antibiotics. Penicillin would probably be the best known of those. But we do have some fungi that are bad for us, of course. The vast majority of parasitic fungi, or pathogenic fungi, are a problem for plants and not animals, but there are some that affect animal cells. For instance, athlete's foot is caused by a fungus. And of course, the plant pathogens are problematic when it comes to crop plants. If you're immunocompromised, that makes you especially sensitive to fungi that would not bother other people. In addition, we have fungi, molds, that are involved in food spoilage and can also cause some rather serious allergic reactions. I've put hallucinogenic drugs on here a couple times. I'll let you decide whether those are good or bad. We can classify fungi into 3 informal categories based on how they grow. So yeast consist of single-celled fungi. Molds consist of fungal cells that grow end to end in a long chain, and then those chains are arranged somewhat randomly and loosely. This is what you see when you leave something in your fridge too long, that fuzzy growth on an orange, for instance. And then we can have fleshy fungal growths. These are also known as fruiting bodies, puffballs, mushrooms, toadstools, etc. These are sexual structures. They're structures that produce spores. Fungi that produce these fruiting bodies are not infectious. A chain of fungal cells connected end to end is known as a hyphae. So what happens is we have one cell that divides and the 2 cells don't separate. And then those 2 cells divide and they don't separate, etc. And we end up with a chain of cells. In septate hyphae, the cells are divided by a cell wall. However, we can have coincidic hyphae where the nuclei divide and the cells elongate, but they never lay down a cell wall. And what we end up with is a long continuous tube that may contain hundreds of nuclei. And the cytoplasm is just freely exchanged along this tube. Recall that the vast majority of fungi are decomposers. They live in the soil and they secrete exoenzymes. Exo meaning outside. So the enzymes leave the fungus, they go out into the soil, they break down organic matter within the soil into simpler nutrients that can then be absorbed into the hyphae. And the hyphae are pipelines. They're tubes made up of cells that may or may not be divided from each other by a septum, by a cell wall. A mass of hyphae is known as a mycelium. So in this one diagram here, you're seeing a mushroom. This is a reproductive structure. And basically what's happened is we have hyphae of two different mating types within the soil that have come together, wrapped around each other, formed this fruiting body that will give rise to spores. So the mushroom is simply a structure that's involved in producing spores. It's involved in sexual reproduction, but the vast majority of the mushroom is underground. Now we do have some fungi that are parasitic, and you're seeing that at the bottom. So at the bottom, you're seeing a fungal hyphae that is invading plant cells. It's got these structures coming off known as haustoria, and the haustoria are going to penetrate into cells, and once they're in the cells, they're going to secrete exoenzymes into the cells, break down the cells, absorb the nutrients, and eventually destroy the cells. Also at the bottom there on the right, we've got a really interesting fungus that has this little trap-like structure that will entice and catch a nematode. A nematode is a little tiny microscopic worm in this case, and it strangles that worm and then it fills that worm up with these haustoria, breaks it down, and absorbs it. So there are some really interesting fungi that actually hunt for little critters. A mold is essentially a shapeless mass of hyphae. And hyphae, again, are chains of fungal cells. Molds can reproduce asexually. They can produce spores that are released into the air without sex. And they can also form spores sexually. They use different structures for these two forms of reproduction. A very common mold is penicillium, and that's what you're seeing on the orange. So penicillium is something that grows at low temperatures. It does quite well at lower temperatures. It's something you're likely to encounter in your refrigerator if you leave something a bit too long. In the bottom photograph, what you're seeing are the production of spores by penicillium. Penicillium is kind of interesting in that no one has ever observed it having sex. And believe me, a lot of nerds have watched. They have not seen this happen. Penicillium produces spores asexually and only asexually. You can see at the bottom here on our diagram, we have a spore that is germinating. So it has to land on a suitable surface. And once it does that, it produces this mass of hyphae. And the hyphae that you're seeing in that example are coincidic. There are many fungi that are dimorphic. Di meaning two, morph meaning form, so they can grow in two different forms. What you're seeing here is a particular pathogenic fungus that will change its growth form if you change the temperature. If it's grown at 37 degrees, it will grow as individual cells. If it's grown at a lower temperature, 25 degrees, it will grow as a chain of cells. So hyphae, which will make up a mold form. There are other types of fungi that can change their growth form based on the concentration of carbon dioxide or based on pH. A fungal infection is known as a mycosis, and these can take several different forms. So first of all, we have superficial mycoses, which relate to the surface of the body. So here we're talking about a fungus that is feeding off of dead skin, hair, and nails. It's not penetrating into living tissue, and because of that, patients may not even be aware of this condition. It's not causing pain or discomfort. We have cutaneous mycoses. Cutaneous refers to the skin. This time we're talking about deeper infections. So we have Haustoria that may be penetrating into living tissue. That's going to cause some pain, some discomfort, some itchiness. The patients are going to be aware of this. More seriously, we can have subcutaneous mycoses. Subcutaneous means below the skin. So in this case, fungus has been introduced into deeper layers, probably through a cut. We can have systemic mycoses. And in this case, fungus have been inhaled into the lungs and then have traveled into the blood and have been transported throughout the body. And of course, this is far more serious. Serious, life-threatening fungal infections are unusual in healthy individuals. We're more likely to see that situation in someone that's immunocompromised or someone that is fighting off a coexisting infection of some sort. Now note on this chart, we've got several fungi that are listed as opportunistic pathogens. An opportunistic pathogen is an organism that is quite common. It's found in the environment, it's found on the skin, and it generally doesn't cause problems. But if the opportunity presents itself, it might. So for instance, Candida is a yeast that's found on the body, it's found in the mouth, it's found in the vagina, generally does not cause issues because it's competing with other microbes. It's competing with bacteria. So within the vagina, Candida is competing with lactic acid bacteria. They're producing lactic acid that makes that region of the body more acidic, something they don't like. If something happens to those bacteria, though, then Candida can grow out of control and cause problems. Let's very briefly talk about reproduction in the fungi. And let's use Rhizopus as an example. Rhizopus is bread mold. This is a very common and generally not harmful mold. So we've got a piece of bread there, and notice that we have two different strains of fungus growing on this bread. We have a negative strain and a positive strain. This is kind of like two different sexes, although not quite, because they're almost entirely the same. They differ just in relation to one gene. There's a slight difference in one gene that determines whether we're dealing with hyphae that are negative or positive. But if negative and positive hyphae come together, what they will do is they will fuse. They'll actually produce gametes. The gametes will come together. That's fertilization. We get a zygote. The zygote immediately undergoes meiosis. It produces this structure that forms haploid spores, and then those are released. Half of those haploid spores are negative, half of them are positive. This is what happens with mushrooms as well. So I mentioned that a mushroom is mostly below ground. Well, we have negative and positive hyphae traveling through the soil. When they meet, they come together, they wrap around each other, and they form a mushroom, and they form spores. Fungi can reproduce sexually or asexually. So what you're seeing in the upper right is sexual reproduction. We have these masses of hyphae. Some of them are negative, some of them are positive. These two mating types are coming together, and where they join, they're jamming a whole bunch of cells together to produce zygotes. That's all happening within those big black balls. Down in the lower left, we have asexual reproduction. So fungi can also produce spores through asexual reproduction. This is just one mating type, just doing it all on its own. And there are some fungi that have totally given up on sexual reproduction. They only do the asexual stuff because it's faster and easier and not quite as messy. Penicillium would be an example of that. One other thing to consider when we're talking about fungi is allergies. There are a large number of people that are very allergic to molds, allergic to the spores that they produce. Black mold is especially harmful to most people. I used to work as a contractor, actually. I used to do a lot of tiling and plumbing and drywall and stuff. And one of the things I got to do quite often was to rip out drywall and get rid of the black mold. So what you're seeing here in the photograph on the right, someone has removed the drywall and apparently the plumbing has been leaking. This has been a moist area, which mold quite enjoy, and they've grown very well, and they're releasing lots and lots of spores into the air, which can make people quite sick. Now, if we have a condition like this, it's actually not that difficult to clean up. You have to remove everything that's wet, you have to dry it out, and then hit it with a lot of bleach. Bleach is actually pretty good at killing most things, including fungal spores, and that's the reason that we use a 10% bleach solution in the lab. If you want to test the air quality, what you can do is you can get an air sampler like you see in the other photo there. And what this will do essentially is it will suck in air. So it's got a fan that will draw in air, and then the air passes over a little plate, kind of like a little petri plate, on which spores can land, and then that plate can be incubated to see what's present. But the toxins that are contained within these spores can be very harmful. They can cause allergic reactions that can be quite severe in some people, and they have been linked to fatigue and depression and other symptoms. So fungal cells can grow in chains, and that's what we see in the molds, but they can also grow as individual cells, and again, we call that a yeast. So the term yeast covers a lot of different species. We're just talking about the growth form. In the photograph on the right, you're seeing yeast that are reproducing asexually. They're budding. So they're producing these smaller cells that will bud off and pinch off and then become autonomous or free-living cells. Yeast, of course, are quite helpful. We can use them to produce ethanol, we can use them to produce carbon dioxide and so on, but they can also be problematic. Yeast infections are referred to as candidiasis or thrush, and we can have yeast infections all over the body, but the most common forms are within the mouth, oral thrush, or within the vagina, which is vaginal thrush. And the most common organism that produces these infections is Candida albicans. I've put together a little summary of the groups that you should know. Also check the study guide that I've posted for additional information. Once again, I've also posted some links to some pronunciation guides that will help you with some of these rather difficult names. Recall that the Protista are a very diverse group of organisms. In large part, this is due to the fact that many of them are probably not closely related. The pathogenic groups fall mainly into the protozoa. These are animal-like protists. They're single-celled, they're ingestive, they take in food in large chunks. And the protozoa move around by cilia, flagella, or cytoplasmic streaming. Cytoplasmic streaming is where a cell produces a pseudopodia, a lobe, and then streams or pushes its cytoplasm into that lobe. Many of the protozoa can form resistant cysts, and that's rather important because that allows them to exist during harsh conditions and also to pass through the stomach, which is a rather harsh environment. Fungi are not Protista. I grouped them together out of convenience, but they're actually more closely related to animals, and we're going to talk about animal parasites in our next topic. Remember that multicellular fungi are made up of hyphae. We have these chains of cells linked end to end. We may or may not have a cell wall that separates those cells. Fungi are classified based on their growth forms, and some fungi can be dimorphic. They can exist as both yeast forms and mold forms. And finally, here's our terminology. I've included a list of featured organisms. We will come back to these again and again, and we'll expand upon this list. And finally, here's a few study questions to share with your closest friends.